I'm Calestas Juma, the director of the Kennedy School's Science, Technology, and Globalization Project. Welcome to the forum. Allow me to share with you how this afternoon's event will proceed. First, we'll hear Dr. Craig Venter's address, Reengineering Life, Implications of Human Welfare. Then we will hear from two other respondents, Secretary Dan Glickman and Professor Dan Wickler, after which I will lead a short conversation among the panelists here on stage. Then I will pass the microphone to the audience. 50 years ago today, James Watson and Francis Crick published their discovery of the structure of DNA, the double helix, which revealed the secrets of life. They discovered that the structure of DNA, when unzipped, makes copies of itself, which confirmed long-standing suspicions that DNA carried life's hereditary information. The success of Watson and Crick was due in large part to their boundless ambition, impatience with authority, and disdain for received wisdom. Today, 50 years after the publication of the Nature article, we are honored to have the scientist responsible for decoding the human genome, making it possible to read the book of life. Like Watson and Crick, our featured speaker today is known for his rebellious personality, an impatience for big bureaucracies, a restless intellect, a confirmed tendency to defy authority, and a passion for risk-taking. Through his sheer will and determination, we will reap scientific rewards and potentially life-saving medical treatments, as well as improvements in agriculture, industry, and environmental management, almost a quarter of a decade earlier than anyone had expected. This spectacular scientific accomplishment, along with research carried out by Dr. Francis Collins at the National Institutes of Health, has been equated by some to that of the lunar landing. We would be wise to review the steps our guest took on his journey, steps that ultimately led to such a giant leap in science and a significant contribution to human welfare. Dr. Venter, a native of Utah, he barely graduated high school. His attraction was to the beaches of Southern California instead of the books of college, and was later drafted into the United States Navy Medical Corps. Serving in Vietnam taught him indelible lessons. He said, "If you suffered fools, you died." I dealt with thousands of people dying because of stupid government policies. While in Vietnam, he refused to follow orders he regarded as unnecessary, which resulted in 2 stints in the brig. Returning to the US, he entered the University of California, San Diego, and emerged 6 years later with a PhD in physiology and pharmacology. Within a few years, he was at the National Institutes of Health. In 1992, he and Claire Fraser, his wife and collaborator, left NIH and established the Institute of Genomic Research. Then, several years later, he set up cellular genomics. And the rest of the story, as they say, is the future of humanity. We are honored today to have Dr. Craig Venter.
Well, thank you very much for a kind and difficult introduction to live up to in 15 minutes. So I'm going to try and take that challenge by dealing with, in fact, some of the challenge we were faced with when dealing with the expansion of the information with genomics. I had a large lab at the National Institutes of Health, but I found that the structures within our science system were completely inadequate to to allow us to rapidly move forward. I looked around at universities and found the universities were completely inadequate because each discipline is taught as a separate stovepipe type of industry. And for genomics, we had to integrate a broad array of disciplines. So in 1992, I left NIH to form the Institute for Genomic Research. I had a $70 million grant to start a not-for-profit institute. And we were able to immediately hire the type of people that we needed that I could not do in the government. For example, I had a top mathematician, computer scientist, that was sort of the last straw at NIH when I tried to get him promoted. And they said, he's not a biologist. We don't hire those kinds of people here. And yet computing was such an essential component of any forward movement in genomics. So we had mathematicians, we had computer scientists, we had biologists, we had physicists, all working together to put new approaches together. And in 1995, the genomic era officially started. That's when we completed the first genome of a free living organism. And it was done with new mathematical tools. Mathematics was the breakthrough, not some technique in molecular biology. We scaled that up and did a number of additional genomes over the next few years. Some of the key pathogens, such as cholera and tuberculosis, staph and strep, that have driven a lot of new innovations in vaccine and antibiotic development. It's clear the technique could go further. I had a chance in 1998 to take it further with a company, PerkinElmer, offered to give me $350 million to use my method to sequence the human genome. I actually thought the first people to call me from the company were kidding, and I hung up on them. Fortunately, they called back and proved that they weren't cranks, at least, well, that's not what they proved, but they proved they had $350 million. We tried to get the money for our not-for-profit institute, and they insisted on doing it in a commercial venture. But we insisted that one thing happened if we sequenced the genome quickly, that they would make it publicly available right away, which the commercial interest didn't quite understand, but agreed to. So we were doing everything that we set up at Tiger, only we scaled up about 100-fold. We had to, in 6 months, build the third largest computer ever built. The largest two were in the US Department of Energy, and they were being used for nuclear weapons simulations, not for assembling human genomes. So we teamed up with Compact Computer and built, at the time, the third largest computer and the largest civilian computer built. It was a 1.5 teraflop computer. It meant it could do 1.5 trillion calculations per second, but it still took several weeks to run the algorithm to assemble the human genetic code. It was a very unique period. Instead of doing the genome in 15 or 20 years, we did it in 9 months. And we had the excitement, along with Francis Collins from the public effort, of announcing what we've been told is the first ever scientific announcement from the White House in June 26 of 2000. We were in a hurry because the implications of having this data. To others in this field, it was an end point to get the data. That's what they viewed their jobs as. But we knew that genomics would drive a new revolution, and that's what we're trying to do now. I left Celera about 1.5 years ago and have started 4 new not-for-profit institutes since then, bringing the total to 5. Tiger's now in its 11th year of existence and has done approximately half of all the genomes that have been sequenced today. Just before leaving Celera, we sequenced the Anopheles genome. That's the mosquito that carries the malaria parasite. At the same time, we finished the malaria genome at Tiger, and we had the fun thing of having simultaneous covers of Science and Nature as we published the Anopheles mosquito and the malaria genome simultaneously. But that brought to where we have the human genome, the malaria genome, and the mosquito genome. where we can look at variants in our own genetic code for susceptibility to disease. We can look in the mosquito for ways to interrupt the life cycle of the malaria parasite, which other than famine is the #1 killer of children on this planet. And also, people have now used the malaria genome sequence already to try and come up with new vaccines that are being tested. That's part of the promise of the genomic revolution. But at the same time, all that's happening, and we have an increased threat from biological warfare, which is going to be even increasing probably in the future. We have major pharmaceutical companies totally shutting down their antibiotic programs. We have companies absolutely afraid to make vaccines because of lawsuits. So at a time where we need new vaccines, new antibiotics, and everybody I'm sure is aware of SARS, new antivirals, because it doesn't matter whether it's a man-made biological warfare agent or a new emerging infection such as SARS, we need new abilities to try and deal with those. One of our institutes, the Center for the Advancement of Genomics, is trying to deal with public issues surrounding our efforts to sequence the human genome. The #1 issue probably right now is genetic discrimination. Our goal is to have everybody in this country have the ability to have their genome sequenced in the not too distant future. And in fact, I've predicted that within a decade before children are born in a hospital, leave the hospital, the parents will have the option of having their child's genetic code on a CD-ROM. There's obviously a lot of implications for that as it drives us into this new era of preventative medicine that we think can actually substantially lower healthcare costs. But people right now are afraid to have their genetic code determined and put on the internet like my genetic code is. At least that's what I've read. Because of this view we have in our society of genetic determinism. It's part of our language, it's part of our public policies. My favorite headline is from the Washington Post: do women have the Zoom gene because they drive around the Beltway faster than men do? Now, clearly they drive around the Beltway faster than men do, but it tries to convert a complex action down to a single gene, and so much of our language does that. Yet very little, if any, of our complex actions and our life outcomes will be directly attributable back to a single or even a complex set of genes, whereas our medical outcomes can be linked back to changes in the genetic code. There was a recent case of a railroad company trying to measure which of their employees had a genetic basis for carpal tunnel syndrome because they had too many employees that they thought they must be faking it, and they were going to use the genetic code to work out who was faking and who wasn't. I don't know if it's correct to use the term whistleblower for a railroad company, but fortunately somebody put a stop to that. And there was no valid science behind this. There was no basis of measuring genetics to understand carpal tunnel syndrome. It was bad policy. It was bad all the way around. And that's the biggest concern, misuse of this information by people who want to discriminate, not because the science will support that. So that's going to be a huge challenge for our policies going forward. There's a bill been pending in Congress now for a couple of years, has over 200 co-sponsors in the House, but the current administration won't let it come up for a vote. So it will probably come up on the Senate side first. That protects people from employers or insurance companies using our genetic code as a weapon against us. Our other institute, the Institute for Biological Energy Alternatives, is what was in the news yesterday. We just got a nice $9 million add-on to a $3 million research grant. It's the nice direction to go from the Department of Energy, supporting our work, trying to use genomics and microbial systems to see if we can alter them sufficiently to produce hydrogen as a direct result from sunlight under conditions that make it economically feasible. So we're actually trying to engineer the photosynthetic process to alter it so we would have continuous hydrogen production as a direct result of sunlight. We don't know if it'll succeed, but I think with the new tools of molecular biology, there's so many opportunities to change things substantially that shame on us for not trying. Also in this institute, we're trying to make a synthetic microbial chromosome to make what might be the first synthetic species. We think this is going to be important long-term to engineer life forms. In the case of hydrogen production, so the cellular energy will be almost totally devoted towards the process of converting the energy from the sunlight into hydrogen production. And in different microbial systems, most of the systems in the cell, from all the genomes we've done, are devoted to evolving that cell continuously. Every microbial genome we've seen has built-in mechanisms for continuous evolution of that species. That's why every virus and every bacteria pathogen is constantly changing in real time. If we eliminate those mechanisms, we can have stable organisms for synthetic purposes. DuPont has been very successful with modifying E. coli to produce a 3-carbon intermediate cheaper and more rapidly than its chemists can. And I think it's just one of what will be tens of thousands of examples in the future of when we computer design species, have robots build them very quickly for specific purposes, whether it's chemical synthesis or bioremediation or energy production. Obviously, it brings a lot of society choices up for grabs with that type of discussion. And one of our biggest concerns is engineering species for biological warfare. Before we started this research, we convened a year-long ethical review at the University of Pennsylvania that involved most major religions, and they came up with no solid objections for doing this. Basically, all the religions said, it doesn't seem to be in their scriptures, so it can't be wrong. It's a legalistic way to view them, but one of the, including links from the Vatican, one of the people on the panel said, well, if God didn't want us to do it, he wouldn't let us. But that hasn't stopped us going too far in policies from our current government. I think the other implications of this work are just thinking about the major issues we're facing, whether it's energy production, trying to change the health of the public, whether it's through intervening with infectious disease or preventing diseases. An area Secretary Glickman is one of the best speakers on, and that's the area of food production. With 6 billion plus people now on the planet, if we don't use genetic engineering, we won't be able to feed them. We can't feed them all now. So I think genomics and this field will impact almost every area of society in ways we can hardly yet imagine. Thank you very much.
Thank you, Dr. Venter, for setting the stage. You've closed on an appropriate note relating to ethical considerations. And these are probably some of the most critical questions that the public is currently concerned about. I wanted to invite our next speaker, Professor Wickler, who has served as president of the International Association of Bioethics, and also served as a senior staff ethicist with the World Health Organization. He currently serves as professor of population and international health at the Harvard School of Public Health to make some comments.
Thank you very much, and it's an honor to follow Craig Venter, who has given us so much. This is an enormous subject, so I'm just going to pick a few of the many topics to say a word about, and I know that you'll raise the ones in discussion that really interest you the most. Let me say at the beginning, because my background is actually in moral philosophy, that the question of coming up with ethical guidelines or an ethical evaluation of the future of genomics and of genetics has a special problem that most other investigations of this sort don't have. When we try to come up with moral theories that would guide us in our thinking about right and wrong, we sometimes turn to what may be metaphors, but also maybe biologically rooted notions such as human nature. Or else, as in with Professor Rawls, we turn to a metaphor about people gathering at the sort of the dawn of a society or in a special place where they're blinded to the social conditions prevailing outside, and they're asked to write the rules that would apply for everyone in that society, including themselves, and they're not told what roles they will eventually play in that society. So we resort to these metaphors and these theoretical constructs in order to generate rules which we then apply to a social problem at hand. Now that's fine, we'd like to do that in genetics too, but the problem here is that if we're able to change human nature eventually by rewiring or reconstituting the human genome, then you can't refer to a fixed human nature that would generate your theories, because really that's what's in question. And one could raise the same issue about these people in this original position of Professor Rawls. Who are these people? What is their nature? What would they think like? What are their interests? All these things are what's up for grabs, and that's what we're trying to figure out. So can we use this instrument, which assumes a fixed quantity, in order to figure out how to change that quantity? It's a special quandary. Now, I'm not going to pursue that, partly because I only have a minute left, but partly because also, these are really kind of blue sky questions where we should not fall in the trap of thinking that in 5 years or 10 years, we're going to be having the human genome in front of us to rewire at will, and we can make ourselves with 3 arms and IQ of 1,000 if we wish, and it's just up to us to decide what it's going to be like. We have problems in the near term that will probably last us, according to the geneticists that I talk to, the rest of my lifetime and probably a lot of your lifetimes too. So we should, while at the same time that we're focusing on these very long-range blue sky questions, we have to ask what kinds of issues are going to come up in the near term. Let me just mention a few. You're all familiar, I'm sure, with the phenomenon of the fetal monitor, which is used at birth to detect stress in the fetus. A great idea. The problem is that it tells us so much, and we feel impelled to act on everything that we find out from this monitor, that we overreact. And it's not clear whether the mayhem that's caused by overreacting to what is actually insufficient and insignificant data outweighs the good that comes about by reacting appropriately to useful data. Now, I say that even though my own child was actually saved by one of these things, so I know they do work. Now, in the case of genomics, we have something similar. There are tests now that will tell you whether you have this or that gene. What do these tests tell you? Sometimes they don't predict very well. Sometimes this gene is one of 35 genes that may have to work together to produce a certain kind of problem. So what do we do with these tests? What happens when these tests are ordered when there's no therapy that would possibly affect the syndrome that the tests are tested for? Or when somebody else orders the test for you, as parents do with children. We don't really know, but we're bound to react. Once the tests show up, once we can read these genes the way we can read Craig's genes on the internet, and someone ventures a guess as to the significance of these, we have to react to that information. Will we react appropriately or not? We don't know yet. There's a significant risk here. We certainly know, as Craig mentioned, that in the case of so-called behavioral genetics, and with genes that are thought to have some impact on behavior, there's a notable tendency to exaggerate, to imagine there's a specificity which is absolutely lacking. The gene for shyness, the gene for boldness, the gene for this, the gene for that appear in the headlines only to be retracted a few months later. You don't read that in the headlines. That happens over and over and over again. Now, as we get near the time when we imagine that a child will go home with his or her genome on a CD-ROM, and you can put that CD-ROM in a computer and various things will light up and it will say these genes are thought to have behavioral significance and here's what the possible significance is. You can imagine how a parent would react, or a girlfriend. So we do have to worry about this. Our reaction can be a lot more damaging than the information can be useful. Another thing we have to worry about is confidentiality questions. Craig also referred to these. These certainly happen on the individual level. There's a real possibility, absent this law, which is coming through Congress, and it is a problematic law, as most laws are, there's a real possibility that the information that you would get back by having a doctor look at your genome and tell you what's there would be a lot less helpful than the damage that would be done to you if an insurance company got a hold of that same information and used the very same data to deny you insurance. Because that doctor will then say, boy, you have big problems here, but adios, I'm not being paid to take care of you. So then you have to go through the back door of the hospital and beg to get in. So each person, especially in a country like the United States where we don't have a common pool of insurance and where bad information about somebody could actually knock them out of the insurance pool, each person is going to have to worry about the insurer getting hold of the very information that the doctor wants to use for therapeutic purposes. Now, when this happens on a social scale, on a society-wide scale, there's a special reasons for concern, and it is happening. Many of you know that the island in the Atlantic called Iceland has come up with a really marvelous data bank in which 90% of the population is now enrolled, and they're apparently on the verge of making some major discoveries. In Iceland, they took very stringent precautions to make sure this information wasn't used. Other countries saw what was happening in Iceland, and they thought, we've got genes too, and we can make a lot of money just like Iceland can. Now, they've rushed to copy them. In the case of one country I won't mention, they took money out of an already underfunded primary care system to put it into this. Is that ever going to pay off? We don't know. Another country, the gene pool was essentially sold to a football magnate from a neighboring country who thought that, who had also invested in diamonds, and he thought genes are the new diamonds. So he sort of bought a license to look at all these genes, and when the information reached the people of the Republic of Tonga, or the Kingdom of Tonga, there was rioting in the streets. So we just don't know what the future is going to be of this kind of social venture, and yet this is racing ahead. So who's minding the store? Who's governing here? No one. We have some governments, but government action. In this country, actually, the government has given a fair amount of money for at least academic studies of these things. But it gets all mixed up with, with the religious questions about abortion and embryos and all that stuff, making sound, sane political action very difficult. And in countries that don't have the infrastructure that we have for public policy and science, countries that are very poor but where some of these same questions come up, there are real problems here. And we can't look to the international agencies. UNESCO, if I can speak as someone who had worked in the UN for a while, UNESCO has come up with a document on this which, if you read it, I think you would, let's just say it's a good thing it isn't law. And WHO, which I think has a sounder approach to this, has the following number of genetically qualified staff: 1. So we are facing a brave new world in which this information has potential gigantic impact. We're not being led well, and it's very hard to say what the impact is going to be on the short or the long term.
Thank you. The question of who governs is Secretary Glickman's domain, I don't have to introduce you except to say that you're familiar both with making laws and hot dogs. Yeah. Having been Secretary of Agriculture.
Sausage, remember, you're not supposed to watch laws or sausage being made, First of all, most members of Congress, most members of the executive branch are fairly scientifically challenged individuals. And at the same time, somebody does kind of have to mind the store here, as you say. And in our system of government and democracy, it's Congress and executive branch that is there minding the store. And it's difficult when you have this very complicated interface of science and policy. I recall when I led the U.S. delegation of the World Food Summit in 1996, and I thought I was doing a wonderful job, and then a whole group of environmental protesters stood up totally stripped naked and threw genetically modified soybeans at me. Written on their bodies were the naked truth and no gene beans. And in talking to them afterward, it didn't really matter what the benefits were. They were against it for ethical or moral or scientific or environmental reasons. And it hit me. These are very complicated subjects. But I think we do have to ask ourselves, one is, what is the role of government here? How do you educate policymakers as to what's going on so that people will never be as smart as Craig is, but they have some remote idea of the issues here, and so that the public is not kept in the dark, which is also very important, because if the public is kept in the dark, which in some sense I think they have been in the agricultural genetic engineering area, it causes all sorts of cynicism and frustration. But the government can be a facilitator, it can fund, as it's obviously done with Craig, and it can be a catalyst. It could also restrict and retard development through tax policies and a variety of civil justice-related issues. There are a couple of issues that I think the government will constantly keep its hand on here. One, which was mentioned, is the area of genetic discrimination, but I more broadly refer to that as privacy. If there is any kind of ethic that runs through this country particularly, it's the desire to be left alone. And you got to ask yourself, how will the policymakers deal with that desire to be left alone, not to have somebody watching them, whether it's the government or their insurance companies, and yet be able to develop technologies which extend their lives and make their families have a much better existence. The other area, of course, is risk management and insurance. These industries are very, very powerful, and I suspect that they will be at the policy table working with policymakers and legislators to deal with the ramifications of genomics. The third area, of course, is religion. Well, Craig mentioned that he didn't know if the major religions would intervene in these issues. I'll bet my life they intervene in these issues. I bet you there are Reverend Falwells and Reverend Robertsons and others in more mainline religions over the next 20 or 30 years who see great ramifications in the moral issues of the human beings acting, quote, like God. And this is going to be tough for policymakers to deal with, particularly with very strongly held views by constituencies who believe in these things very, very passionately. And I've seen it in the stem cell debate. You've seen it in the abortion debate. And there are a lot of parallels. They're not exactly the same, but there are a lot of parallels in this. The final thing I would say is if we don't go down this road, I think that, you know, we will be certainly missing out on enormous benefits for human health, environmental health. I know in the agriculture area that I was involved with, most of the work that was done certainly before the 1960s was done on animals and plants. I mean, that's where all the genetics was basically done. And it produced a miracle of technology and productivity. And of course, it just scratched the surface. Now we're starting to get into things that really do make a difference. What the folks in agriculture did not realize is that if they couldn't relate the products they were involved with to something that actually benefited people, like making you live longer or healthier, then it was tough to get the public very interested in this because they weren't interested in products that helped chemical companies be more productive. They were interested in products that actually did good things to you, like made you live longer, made us grow hair maybe, or do other kinds of things that might be useful to us. And I think the big trick is to ensure that these very, very difficult issues, practical issues, legal issues, moral issues, we find a way to get them into the policy arena in a sensible way so that we can act rationally as a nation and as a world in dealing with the advancements of science, which we so desperately need.
Thank you, Dan. I think I'll forego my questioning and just hand over the mics to the audience now. There are 4 mics over here, 2 down here and 2 upstairs. If you could just give your name, your institution, and
Hyman Hartman
keep
at
it
MIT. The question is, what happens if genomics, especially human genomics, leads to nothing? Let me give you an example. We have the sequence of the AIDS virus. It's a rather small virus. It's got about 9 proteins. And we've known the sequence for a long time. We know every single protein. And I don't think we've had much impact on AIDS. The second point I wanted to make was more with respect to agriculture. And the second point is that we are throwing away our seeds for the companies who are introducing genetic engineering. And the greatest danger right now of genetic engineering is in agriculture. Monsanto tried to put into its seeds a death gene so that you could not use the seeds after one planting. So I think the greatest danger of genetic engineering right now is who is going to be able to control the food.
I think the first question was rhetorical, so I'll let you answer the second.
It was not rhetorical.
Well, on the second point, you know, obviously we had a problem with a gene that was called the terminator gene, which was unfortunately described. But my guess is that these are policy issues, intellectual property rights, who owns these property rights, and how you preserve a diverse system of production agriculture when you have a changing way of developing technologies in production. I just hope that policymakers can address those things and preserve a system of agriculture where we don't end up with a feudalistic system where one or two people own it all. And I suspect we can if we put our minds to it.
Just to be fair to Monsanto, their argument was that no one was making the farmers buy the terminator gene affected products. Farmers would buy this if they thought it was advantageous to them to buy it.
If the first question wasn't rhetorical, then let me at least attempt to give them an answer on it. I assumed it was rhetorical because, you know, the assumption that getting more knowledge on itself is not an important thing. If we can't cure disease with it, it's not worth the time or money to do. Obviously, most of the people here would reject that type of notion just because the AIDS virus is too complex for us to understand in its simplicity right now doesn't mean the knowledge wasn't worth obtaining and we won't be able to understand it in the future. We can understand systems such as microbes quite well, and in fact, working with Chiron, trying to do something about meningitis, within less than a year of sequencing the genome of the meningitis genome, there was a new vaccine ready to go into clinical trials, and it's undergoing clinical trials now. So I think the notion that something might not come out of it of practical benefit is somewhat not worth worrying about if we increase our basic knowledge, even if it takes 50 years before we know how to truly understand our own physiology. It doesn't matter to me if a cure doesn't come out tomorrow. This is true basic science that we're going to drive forward.
Over there and then down here, please.
Abby Hafer from Mount Ida College, and I'm fascinated by the energy question that you've been talking about as regards microbes and hydrogen. This strikes me as about the most exciting thing we could be doing right now, since it's about the best thing I've seen as an idea for finally getting over the CO2 hurdle. Assuming these microbes aren't going to be producing carbon dioxide, we may finally get a handle on greenhouse gases. My question is, since you're already doing this fine work, and it sounds from what you said as though you were hoping to have the microbes do essentially photosynthesis, using sunlight as their energy source, which sounds great, and obviously the sun's a renewable resource, more or less, but you could hit a home run if they did fermentation of solid waste instead. Is there any thought of looking at that?
I have to do one miracle at a time.
Yeah, I know, but you're good at it, you know, so thought I...
That's a good answer, isn't it?
I think we've been trying with what our small institute's doing to raise the visibility of this entire area, because in fact, none of these ideas are new.
Right.
People have been looking at the potential for microbes to produce hydrogen for 30 or 40 years. The problem is the existing systems don't have sufficient metabolic activity to do that on the scale we need for economic success.
Sure.
So what's new is now we have genomics, we have modern tools in molecular biology where we can go back and look at these same questions again to see if we can use these tools to modify things sufficiently to do that. And I think the fact that the government stepped up first with the initial grant a little less than a year ago and then supplemented with $9 million yesterday is an indication that there's more attention being paid to this. And we're hoping not only will our small institute get more traction on this, but it'll raise the visibility enough to start looking at the use of modified and non-modified microorganisms for bioremediation purposes, for all kinds of either CO2 sequestration or energy production. The trouble is we know less than 1% of the microbial repertoire on this planet. So the other thing that we got money for was our notion of applying all the mathematics and computing and whole genome shotgun tools to the environment. And so we're actually doing a shotgun sequencing of the Atlantic Ocean, starting with the Sargasso Sea, where we filter large amounts of seawater through filters, collect all the microorganisms, isolate the DNA en masse. We're sequencing it and using our computers to deconvolute that into the 99% of species that we have no tag on whatsoever. So we're turning biology on its head, where we're going to, for the first time, for most species, see their genetic code first and use that as a probe to actually pull them out of the environment, hopefully finding new pathways, new photosynthetic pathways, new CO2 capture pathways in the process.
Yes, please.
Thank you. My name is Bill McCamley. I'm a second-year policy student here at the Kennedy School, and my question is one based on timing. It seems that the United States government, at least our government, was set up to move slowly. The Clean Air Act took 7 years to get through Congress, and society-wise, religious-wise, a lot of these questions could take years, decades, millennia sometimes to even talk about. Yet the human genome was, like you said, uncoded, decoded in 9 months. So my question is the scientific process moving too fast for the government and religious, societal areas of our civilization to keep up with? And if it is, what can be done? I mean, to slow down the process or speed up the other?
You are the philosopher. How do you speed up time?
Well, I'll leave that to Craig, but one thing here is I think we should distinguish two things. One is, how can we sort of get wise about the ethical issues? And the answer is, gather the evidence, have symposia like this, think very hard. And we can do that prospectively. When Watson got money for the Human Genome Project from the public sector, as many of you know, he in the congressional hearing, he said, look, I'm going to spend $3 of every 100 you give me on the ethical, legal, and social issues. And that impressed the congressman very favorably. A lot of work was commissioned to try to figure out what the impact would be, and even some philosophical work. I co-authored a book using some of that money, which I was very happy to get. So this kind of consideration is going on with the Genome Project more than any other scientific project, I think, in history. It's really a remarkable development, and the American initiative was copied in Europe and Canada, even in Japan. So that's one thing we can do. The other question is whether some of these events run afoul of religious, strong religious feelings, or, to put it a little bit more cynically, whether people who want to score some points with a certain religious group can seize on some of these developments and play games to stop things and then get votes. Now, that's, I think, a very different phenomenon, and there's absolutely nothing that we can do in the academy that I know of to head that off. It's just, it's unfortunate when something that has the promise of a tremendous amount of benefit gets caught up in political games. But that happens, and I guess we have to ask either politicians or political scientists how to avoid that.
Well, I'll comment just very briefly. I think it's a wonderful question, and I don't think science is moving too fast. I think the dissemination of the information and the understanding of it is probably the most critical issue. I think a lot of the, quote, ethical issues that were raised, half of them are pure science fiction, in my view. And the challenge in making any policy is sorting out what's real, what's likely, and what's just fiction. On the other hand, our government, in my view, is set up to move as slowly as possible by design, so one person, one party can't come in and radically change the system. So it's designed to be sluggish, and it fulfills its design very efficiently.
We have a question up there, and then I'll come down to you.
Hi, my name is Josh Laxina, and I'm a senior at Harvard College. My question is primarily directed at Dr. Venter. I'm training to be a scientist as well. And recently with SARS and with the Human Genome Project, scientists have been thrust into the public spotlight and have had to interact a lot with politicians and policymakers. So I wanted to know what you feel the responsibility is of scientists to help the politicians make better decisions. How can scientists reach out from the ivory tower of their laboratories and try and get more involved in public policy processes? But I also wanted to hear from the political standpoint what sorts of strategies you would like to see implemented to get more interaction between the two worlds.
I think it's a great question because I think every scientist has a public responsibility, whether they're living off of public funds or private funds, to speak out about what they're doing and what they think the data and what they're doing means. But part of our scientific societies discourages that. You know, if scientists end up in the media too much, they get attacked simply for that fact alone. Where if it was the standard fare, if people were speaking out, trying to educate the public, perhaps we'd be better able to discriminate between what's fact and fiction. So it's critical. There's, one physician right now in Congress. There's no scientist, to my knowledge, in any part of the administration and at the senior levels. The White House Science Advisor's Office is not a major source of information and knowledge, even for the politicians. We got rid of OTA, the office in Congress that actually made technology assessments and educated the Congress about issues. So we're left to individuals speaking out. But we have a very large number of people doing this work that probably only a fraction of 1% do any public speaking on it at all.
Okay, if I just make, when Napoleon said, two prongs to this. One is I agree totally with what you said. There is no science background there's some congressional staff with some, but in the halls of Congress itself, it's very limited. And except in the Pentagon, in DARPA, and to some degree at NIH, the science establishment has not advanced very far in most of the other government agencies in terms of, and that's just something that requires the commitment, funding, and resources. Another thing. The other thing is on the other side, Napoleon said war is too important to be left to the generals. You know, if all you are is talking to yourselves all the time, then you don't tend to think anybody else is important. And most of the American public are interested in these issues, and they cannot be taken for granted, because if they are, they're taken for granted at the peril of the science community.
Yes, thank you.
Hi, Daniel Collins, MIT student, Pugwash. At the end of the opening speech, you commented that there are many things unanticipated that may arise from genomics. It was put in the context of many good things, and certainly many good things have come, and many good things will come. Earlier on, you mentioned shame on us for not trying to employ these techniques to improve quality of life, but surely also shame on us for not trying to temper our lust for development and trying to filter out the adverse effects that may arise. So what role of scientists in the choosing of what technologies to go forward with that may or may not end up in bad consequences?
Well, you're absolutely right. We have a responsibility to do both. We try to set a new precedent before we set out to do the engineering of a chromosome in a cell. As I said, we had a year-long review of this process, and until that process was completed and said they thought it was very reasonable for what we were doing and how we were doing it to go ahead, that we went ahead. I think this is now being viewed as a new paradigm of how in some of these more complex issues to start with the discussion first, followed by the science. Sort of deals with the question over here, are we going too fast? I don't think we're going too fast, but it's not wrong to consider the ramifications now in this era, not only of modern communication, but where you can have a disease breakout in China and a day later have that same breakout in Toronto, have information on the tools of this field be used deliberately for sinister purposes. We have to take that into consideration, and I think that needs to be part of the discussion of how we go forward and what should be done. I'm not of the philosophy just because we can do it, it should be done. I think human cloning or stem cell modification is, in terms of germline modification, There's no rationale for doing that right now. Number one, we don't have the knowledge to do it intelligently, and sex is pretty darn good.
Thank you. My name's Dr. Ruth. What a point to try and interfere with a simple question. My name's Dr. Ruth Rabinowitz. I'm a member of Parliament from South Africa, and I'm also a medical doctor. And I sympathize very much with the people who are saying not enough money is spent on strengthening members of Parliament in terms of their understanding of science. And I'll precede what I'm saying by saying I'm very jealous of you because I'm involved with NGOs where we try to raise money to cope with the problems of life. You're being given money to change the way life works. And on that score, you're doing what the man who was the first one to clone Dolly said, we are embarking on the second creation, and you are, in a sense, on that road. But I'm a bit concerned about the casual way in which we talk about the monitoring. We don't want government to regulate because then they select what science they want to promote, military things or government agenda. But what are the people who are on that road to the second creation, the genetic engineers, the cloners, the genome people, doing to filter their processes themselves, to see that we have an overview still, that we can see how we're damaging the ecosystem, that there is a balance. One only discovers now that the FDA, for example, didn't have stringent controls on people who did genetic engineering of foods. They're only starting to implement those now. And one can do that too late. So what are you actually doing yourselves to filter your procedures and also to catch up to where you've gone, because science has gone quickly so that, for example, we still have abnormal babies being born and we have people with Down syndrome and we don't seem to know simple and uncomplicated ways of picking that up. But we seem to know how to be very remotely technical, very fine-tuned technical without an overview, without a holism about it.
Should I ask the parliamentarian?
No, I think that's your question.
I could answer it.
Well, you did owe me one, right?
Yeah.
But well, let me use one of your examples. Down syndrome is a trisomy of chromosome 21. That is absolutely detectable at the early possible stages. And most modern clinical centers do screen for that, but they usually only routinely do it if women are over age 35, as an example, unless it's requested. So a lot of these things are testable for and preventable if you accept abortions as prevention. In terms of the complexity of understanding what causes that extra chromosome in the first place, we could be light years away from that, we could be 2 years away from it. We don't know. And so understanding the mechanisms, I think that was one of the biggest shocks to come out of the human genome, is people were expecting, in many cases, 200,000 to 300,000 genes, because they thought our biology worked in a linear fashion. There was going to be one gene for every function. I've argued the biotech industry took it even further. It was going to be one gene, one protein, $1 billion. They were all in shock when we found 26,000 genes, which meant there wasn't going to be a linear function in terms of genes and outcomes. The pharmaceutical industry almost stopped cold using genomics because it wasn't going to provide what they thought. It means we have to understand how 26,000 genes produce 300,000 proteins in 100 trillion different cells. That's why I think a lot of these things are science fiction for now. It's going to be 10 or 20 years before we have a computer large enough that can model how we go from a single cell to 100 trillion cells with 300,000 different components. So all these people, the comments worrying that science is going too fast, it's not going fast enough. It's going to take most of this century to understand our own biology, and 99% of what's going to be discovered remains to be discovered. I think we should go faster, but dealing with the discussion for the ethical implications as we go.
Can I just throw in something? Your choice of this example leads me to call attention to something that may sound at first quite different. Just this exact case was seized on for the opposite reason by a number of people who are worried about the ethics of genetics. An economist took a look at the economics of testing women with amniocentesis and other tests to catch the cases of Downs. And according to these economists, although the hit rate for women under 35 is much, much lower, which is why they're not currently tested, the cost of caring for someone with Downs over a lifetime is so great that you'd actually save money if you tested absolutely every pregnant woman who would agree to be tested. And they published this, and they said that basically what they were saying was that a universal testing program is cost effective, which would take care of the problem that you're concerned about. But then other people said, we have eugenics again. What we're deciding is we want a world without people with Downs. And so we're going to ruthlessly go through the population and make sure no people like that are born. And so they took this as a sign. The thing that you're saying would be a solution, I think, was taken as a danger sign that we've reached a dangerous point with genetics.
Okay, could
I'm Evelyn Fox Keller. I'm from MIT, and I have a question for Craig. And that follows on the previous questions. It's really more of the same. But it's focused in particular on your program to engineer new life forms. And you mentioned bioterrorism, the risk of bioterrorism, several times in your talk, but I didn't hear what you're doing about how you're dealing with that problem, that risk, and how much thought has gone into the real dangers of the biotech industry replicating the computer virus experience and the transmission of the technology everywhere.
Well, it's actually a question I've given a great deal of thought to. In fact, I'm the one that introduced the previous administration to the issue and the concerns about smallpox. Because after I sequenced the smallpox genome and found how close it was to the vaccinia genome, I argued that within a decade, somebody would be able to just to synthesize the chromosome for the smallpox genome. And that got the Clinton administration moving, and that's why there's in fact been so much concern in part on vaccination against smallpox. It is a serious issue. It is a serious, at least potential threat. And it also helped that Richard Preston wrote a great book that dealt with a modified version of smallpox in New York that got people's imagination going. We actually postponed what we were doing in this area primarily because we had to have a solution that allowed us to go forward and yet deal with what I consider a very real potential threat in this area. There's on the order of 20 companies that synthesize oligonucleotides right now. And the case that was published, which I attacked as almost a prank, the poliovirus synthesis, was done by ordering a large part of it just from a synthesis company. So it's a real problem now, but going to things that are 100 times larger, we decided that we have to undergo some form of self-censorship in this environment. So we indicated, and it's quite controversial, that we may not disclose all the methods that we are developing that allow us to synthesize something 1 million base pairs long instead of only 5,000. But because one lab that's dealing with the ethical issues doesn't do it, everybody was shocked by the poliovirus being published, that somebody would deliberately make a human pathogen and show the world how simple it was to do, I thought was absolutely astounding. So it's a complicated issue. There's no absolutes with it. But just because I don't do it doesn't mean it's not going to go forward as a field.
Well, I'm glad to hear that you've given so much thought to it and are attentive to the need for self-censorship, but you're asking the public to invest an extraordinary amount of trust in your ability to foresee the problems. Don't you think that there should be a larger oversight of future scenarios and risks?
Before he answers that, the answer is absolutely yes. Whether that oversight should come from a RAC-type committee, let's hope not. I think what we need is to ensure that we have the ability to deal with biological warfare of better vaccines, better antibiotics, and new antivirals, and the government should be supporting the solution, not trying to block science from going forward.
Dan, do you have a
No,
comment on
no,
the
no.
regulation? Let's go over there, please.
Afternoon. My name is Derek Lewis. I'm from the business school. Seems like there's a lot of terrific basic research going on. To me, the big question is, how do you make sure that that value is captured over the long term for the public? And a lot of what I've seen in the biotech space has to do, obviously, with commercialization of the advancements that are created from the basic research side. I guess my primary question is this. In Arizona, where I come from, the tech transfer policy is quite different, for example, than some of the other tech transfer policies that exist in other states. And it seems that in the U.S., differences like this make it more or less difficult for given regions to try and capture value that's being created in the laboratory and put it out into the commercial sector to build businesses. What would you say are some things that ought to happen either in partnership or independently between the private and public sector to try and essentially optimize our ability to capture this value and commercialize the basic research into businesses that can go on forward? What kinds of things need to change in the public sector in that sense?
I actually see the problem is at the other end. I would ideally like to see, if there was one change in government, it would be to substantially slow down the patent office and what they consider patentable in this field, so that real discoveries are what drive forward commercialization. It's great to have drugs patented. I think what set the field back was starting with the federal government and then rushing into biotech and pharmaceutical companies trying to patent every gene that I and other people discovered. So I think some of the patents that are being issued right now by the patent office are just extraordinarily stupid. And I'd much rather see we show some restraint there while we develop the proper policies. And if somebody has a real product, they have a chance for getting it commercialized.
Get over there, please.
I'm Jamie Staples. I'm a first-year PhD student in the health policy program here, and I'm concentrating in ethics. I'm interested in knowing, first of all, I'd like to say that I'm very glad to see the three of you on a stage together discussing this issue. But I'm interested in knowing what you feel, in light of the fact that so many of you have said that you feel that there's room for some sort of government involvement. What is the role for government in this as far as regulation of genetic advancement, scientific advancement? What is government doing right now to stay on top of the advancements and to sort of consider the sociological ramifications of these things? Are you keeping up with what's going on in the science? And if so, what role do you feel that you have in regulation? And as a scientist, what role do you feel government has in regulating what you do?
Dan?
I can't speak to all government. I would say in the agricultural arena, where the bulk of this work was done up until maybe 20 years ago, the regulation was pretty primitive. And there is still not much of an infrastructure. There are a lot of different federal agencies with a lot of different acronyms trying to struggle with this. And you also have a lot of turf battles between agencies. And then the issue is spilled over into the international arena, and the politics of trade has engaged in it as it relates to agricultural biotechnology and genetic engineering. But I think there is the recognition that the government, to the extent that they're going to be involved in regulation for safety of the public, whether it's food safety or environmental safety, I mean, public safety is the dominant theme of the regulatory agenda. And I think there is a bipartisan recognition that we need to upgrade the systems that we have, the infrastructure of the systems. Unfortunately, it's been very hit and miss, and it's been rather haphazard, and it hasn't had high policy level interest at the White House or at the congressional level to date. But I mean, it doesn't mean that it's non-existent. The public is demanding this, and the NGOs and the interest groups are demanding this, and now the industry is demanding it as well, because if they don't have public confidence in the research or the products that are coming out, they're dead economically. All this research is for naught. So, but it is very, very slow, and it's not very well developed, and the science community has not necessarily been extremely helpful to the government in coming up with public policy solutions to the problems.
Yes, please.
Yeah, I'm Richard Sobel, and one of the areas I work on is privacy and confidentiality. And I want to direct this to maybe some back and forth between the panelists. Dr. Venter mentioned the attempt to get anti-discrimination legislation through the Congress. I mean, here we're celebrating the 50th anniversary of a wonderful invention, discovery, and your own sequencing of the genome. Tremendous opportunities which are held back because people cannot be encouraged to have certain tests done, because even if there's therapy, the prediction will be that they will have the disease rather than a probability. So the science says one thing. The political process hasn't moved to come up to those sorts of protections, so it can't be fully enhanced. Congressman Glickman mentioned that people want to be left alone, and yet we're seeing that the legislative process often has one purpose: protecting privacy. Everybody's getting HIPAA notices this week. It's supposed to protect privacy, but it's actually disclosure regulations. So my question to the two gentlemen are, Congressman Glickman, how would you advise Dr. Venter and others who would like to see a legislative advance that would help this revolution be effective, get through the Congress, and actually provide the protections that would benefit us all in the long run?
Well, I don't know how I'd advise them, because it's a very difficult problem. But I'll tell you, it's not an impossible problem. Look, we already have non-genetic risk factors that insurance companies can take notice of. You smoke, you're too heavy, you have a pre-existing physical condition. My sister had ulcerative colitis all her life, and so she developed colon cancer, and her insurance company talked about the pre-existing condition in there, and she had to wrestle with that from them. I don't see why you couldn't develop a protection so that the public does not get excluded from health care coverage, but you allow risk factors to be identified so people can properly take care of themselves and help prevent disease. And I mean, that's the role of the public policy process, to do that kind of thing. And I don't understand. I mean, it's not impossible to do it. Whether there's the will to do it or not, I don't know.
But that would not be a solution to the problem, because that would really just allow the insurance companies to raise their rates for the risk factors. Unless you have, as somebody suggested, universal health coverage or anti-discrimination.
No, you would have to have that as well. I'm talking about, you have to have the privacy protection where appropriate, you have to have the consumer protection, you have to have the rate protection, all those kinds of things. But there's no reason, I would hate to see this stop the development of the technologies, because right now we know what a lot of risk factors are for disease, and that's part of the process of healthcare, ratings, and everything else.
I mean, one of the standard cartoons in this area is a car accident, and ambulances and tow trucks arrive. The tow trucks have no problem because there's no fault auto insurance, but the ambulances don't know what to do depending on whether or not the injured people have insurance or not. They have to decide which hospital, if any, to go to, and who's going to pay them. You had the answer. You know, if anything ever justified universal health coverage, it's the human genetic code, because as soon as we can sequence everybody's genome, we'll all be uninsurable.
That's a good point.
I have 2 minutes left, and I would like to squeeze as many questions as answers in those 2 minutes as I can. So let's go over here. And then the last question.
I have 2 questions. My name is Ruth Bellingini. I'm a Brazilian journalist and a Knight Fellow at the MIT. And one of my questions is, how good is a genome sequence without annotation, without identification of its elements and function of the genes? And
That's easy, not very.
Yeah.
Okay.
And the reason I'm asking that, it's for the last 5 years I've been writing and lying to the readers, saying that the second line is, it's going to cure disease. And they believe it, but they're not believing it anymore. Is there any way for people to write about science without making anything a hype, like a race between the guy with a lot of hair and the guy with not so much hair, or big promises that in the short term we'll have big answers to big problems?
Yeah, I think it's an issue that needs to be taken up with news editors. We've dealt with hundreds of top science reporters that would like to write detailed, honest stories, and they go back to their editors and they can't get their stories published, or they get stripped down to the simplistic headlines. So, you know, what gets condensed is the gene of the day story. Scientists do this in part to try and get their next funding or get their biotech company through its next round, but most of it is everybody wants a simplistic story. And guess what? Your biology is not simplistic. It's incredibly complicated, and that's not a story that any news agency really wants to deal with.
Okay.
Thank you. My name is Victoria Daco, and I'm a professor of economics from the City University of New York. I have two quick questions. The first is, do you have an estimate how much money was spent on the genetic research towards altering the molecular structure of the DNA? Would you have an estimate of the amount of money?
I have no idea. The Public Human Genome Project was somewhere between $3 and $5 billion, but that was just a tiny portion of all research that's been going on for the last 50 years.
My second question is whether there is any successful experiment to alter the molecular structure of the DNA permanently.
I think there's been tens of thousands of them, not altering the structure of DNA, but altering the sequence of the DNA in terms of inserting genes permanently. In fact, Monsanto has lots of examples just in the plant area. But this is routine in every molecular biology lab, at every university, and every research institute in this country. It is a standard technique. So it's the whole field of molecular biology. Absolutely, yes.
Could you have the last question over here?
Thank you. My name is Richard Cash from the Harvard School of Public Health, the Department of Population and International Health, and my question is international in nature. As you know, many developing countries are quite fearful of the loss of their patrimony and genetic, because of this whole drive in this country towards the protection of intellectual property rights. How critical is this protection for the development of new innovations? Could it be eliminated? And if not, what do you all suggest to relieve the anxiety of the countries of the world who feel that they're going to either lose out or not be part of this process?
I have a solution that's relatively simple, and that's going back to our early history. At the start of this country, people that were inventors were encouraged to steal every invention they could from England, from Europe, and if they did so, they were rewarded with a patent in this country. That's how we got our start. And only after we had an established system were more stringent controls put on that they were actually new inventions. I think we should model our system for the developing world. And it takes away the issue of ownership. We should encourage every developing company to steal our intellectual property. We should give it to them until they reach a certain level, and then we enforce the next level rules.
I think on that note, I never thought that we would be discussing here theft as a public policy response to these issues. But I wanted to thank you very much for sparing the time to come and share with us this afternoon and our two respondents. And let me welcome you back again.
You're very refreshing. It's a pleasure to see you.